Hello everyone. Today we are going to start an important and interesting area of food and nutrition, nutritional biochemistry. Before we get into the topic, I want you to think about one simple question. What actually happens inside our body when we eat food? The carbohydrates, proteins, and fats that we consume don't simply provide energy directly. They undergo a series of chemical reactions inside our cells, and these reactions need to happen rapidly and precisely for our body to function normally. So, what controls and speeds up all these reactions? That brings us to one of the most important concepts in biochemistry, enzymes. In today's session, we are going to understand the foundations of biochemistry, enzymes, and how enzymes carry out their actions. Biochemistry is the branch of science that deals with the chemistry of living organisms. It studies the structure, composition, and chemical reactions of substances that occur within living cells, tissues, and organs. It explains life processes at the molecular and cellular level, such as metabolism, enzyme action, genetic expression, energy production, and molecular communication. In the context of nutrition, biochemistry helps interpret how nutrients access chemicals in the body and how their interactions influence health, growth, and disease prevention. Nutrition is a science that relates food to the functioning of living organisms. It includes the intake of food, digestion, absorption, transport, metabolism, utilization of nutrients, and elimination of waste. Since nutrients are essentially chemicals and the human body is a chemical machine, nutrition cannot be fully understood without biochemical principles. So the major objectives of biochemistry include to understand the molecular basis of life process such as digestion, respiration, muscle contraction, nerve transmission, immunity, and reproduction. To study the structure and function of biological molecules, carbohydrates, proteins, lipids, nucleic acids, vitamins, and minerals. To explain metabolic pathways, their regulation, and how energy is produced and utilized by the body. To investigate the biochemical basis of diseases, especially those caused by nutrient deficiencies and metabolic errors or toxin accumulation. To apply biochemical knowledge in clinical diagnosis and treatment, such as enzyme tests, blood analysis, hormone assays, and metabolic markers to provide the foundation for understanding nutrition, food metabolism, and diet related disorders, to support research and technological development in medicine, nutrition, food science, agriculture, biotechnology, and pharmacology. In the scope of biochemistry, it has a vast scope covering both basic and applied sciences. In that, the first one is structural biochemistry. Study of structures of biomolecules such as proteins, lipids, nucleic acids, carbohydrates, and their interactions. In metabolic biochemistry, understanding metabolic pathways such as glycolysis, TCA cycle, beta oxidation, amino acid metabolism explains how nutrients are converted to energy or stored for later use. In enzymology, study of enzymes, their mechanisms, factors affecting enzyme action, and clinical importance. In molecular biology and genetics, understanding DNA replication, transcription, translation, gene regulation, and mutations. Clinical biochemistry, application of biochemical tests to diagnose diseases. Example, liver function test, renal profile, glucose testing, and lipid profile. In nutritional biochemistry, study of nutrient functions, deficiency symptoms, biochemical markers, and diet studies relationships. In immunochemistry, molecular basis of immune responses, antibodies, antigens, and post defense. And biotechnology and applied biochemistry, genetic engineering, fermentation technology, drug design, bioproduct development. So, what are all the interrelationship between biochemistry and other biological sciences? So there are interrelationship between biochemistry and nutrition when it comes to digestion, absorption, metabolism, and utilization of nutrients. And in biochemistry and physiology, it will explain how ATP's role in contraction. In biochemistry and medicine, we can understand the disease at the molecular levels. When it comes to biochemistry and microbiology, the biochemistry supports the fermentation, antibiotic production, enzyme technology, and vaccine development. In biochemistry and genetics, the gene expression, mutation, inheritance patterns, and DNA technologies depend on biochemical reactions. So, when it comes to biochemistry and cell biology, uh, we study about the cell structure and function, organelles, membrane transport, and cell signaling. And in biochemistry and pharmacology, study about drug actions, drug metabolism, and detoxification are explained. Biochemistry and immunology, biochemical molecules like antibodies, cytokines, and complement proteins. In biochemistry and environmental science, we determine the pollutant metabolism, toxin action, and ecological balance. So, basically, what are enzymes? So, enzymes are defined as protein catalysts that have great specificity for the reaction catalyzed and the molecules acted on. A catalyst is a substance that increases the rate of a chemical reaction without being permanently altered itself. The enzymes speed up the cellular reactions. The reacting molecules are called substrates, and the substance formed are the products. Many enzymes are indeed pure proteins. However, many enzymes consist of a protein, the apoenzyme, also known as a protein component, a cofactor, required for catalytic activity. The complete enzyme consisting of the apoenzyme and its cofactor is called the holoenzyme. If the cofactor is firmly attached to the apoenzyme, it is a prosthetic group. Often, the cofactor is loosely attached to the apoenzyme. It can even dissociate from the enzyme protein after products have been formed and carry one of these products to another enzyme. Such a loosely bound cofactor is called a coenzyme. Here we can see the two types of enzyme, which is simple versus conjugated enzymes. Simple enzymes are known as proteins only, example pepsin and trypsin. Conjugated enzymes known as protein quotient apoenzyme or non-protein factor which gives holoenzyme. And the cofactors are metal ions such as magnesium and zinc or organic coenzymes NAD, FAD and OA. So the simple enzyme made entirely of proteins yield only amino acids on hydrolysis, perform basic digestive and metabolic functions. When it comes to conjugated enzymes, these enzymes consist of protein portion and a non-protein portion. So in that we have seen the type of cofactors, the metal ions, organic coenzymes, prosthetic groups, flavor proteins, and carbonic anhydrase. The metalloenzymes, where the metal ion is an integral part of the structure, the metal is tightly bound and essential for activity. Examples cytochromes and carboxypeptides. In the isoenzyme, multiple molecular forms of the same enzyme found in different tissues to catalyze the same reaction but differ in their amino acid sequence, electrophoretic mobility, Km value, Vmax values, physiological functions. So it is used as a diagnostic markers. Example, LDH isoenzymes in heart and liver disease. When it comes to enzyme specificity and reaction models, enzyme specificity is one of the most important characteristics of enzymes. It refers to the selective nature of enzymes in choosing the substrate and the type of reaction they catalyze. Each enzyme acts only on particular molecules and performs a specific type of biochemical reaction. The enzyme shows specificity with respect to the nature of the reaction they catalyze, the substrate on which they act. Example, proteolytic enzymes act only on proteins. Urease acts exclusively on urea. So succinic dehydrogenase acts only on succinate. In the types of specificity, we can see the three types: absolute specificity, group specificity, and stereospecificity. In absolute specificity, an enzyme acts only on one specific substrate. Thrombin acts only on the peptide bond between arginine and glycine in fibrinogen. It cleaves exactly one bond to form fibrin for blood clotting. This represents maximum specificity, which is known as absolute specificity. In the second type, group or relative specificity, enzymes acts on a specific type of bond or particular functional group, not on one single substrate. Examples, trypsin breaks peptide bonds only on the carboxyl side of lysine and arginine. It shows limited but specific action. Subtilisin, a bacterial enzyme, cleaves any peptide bond, shows the least specificity among proteolytic enzymes. This shows the spectrum of specificity across enzymes. The third type, stereospecificity. Some enzymes act only on one stereoisomer of a compound. Example, kidney deamino acid oxidase acts exclusively on deamino acids. L forms are not accepted. In geometrical specificity, some enzymes act only on a particular geometrical isomer. Example, difference between malic acid and fumaric acid. Certain enzymes act only on one form, not both. So the basis of specificity is active site structure. Enzyme specificity originates from the shape and chemical properties of the enzyme's active site. Substrates bind to the active site, forming an enzyme substrate complex. Only substrates with complementary shape, charge, and functional groups bind properly. In that, models explaining specificity. There are two types of models. One is lock and key model, and the other one is induced fit model. So lock and key model invented by Emil Fischer in 1894. Active site is rigid and complementary to the substrate. Substrate fits exactly like a key into a lock. It explains high specificity. In induced fit model, it was invented by Koshland in 1960s. The active site is flexible. Enzyme changes shape after substrate binds. It produces perfect fit and allows catalysis. Enzyme kinetics is the study of the rate at which enzyme catalyzed reactions proceed and how factors such as substrate concentration influence reaction velocity. Enzyme increase reaction rates significantly, and this rate can be quantified using parameters like reaction velocity, Vmax, and Km constant. So the effect of substrate concentration. At the beginning of a reaction, when substrate concentration is low, only a few enzyme active sites are occupied. Reaction velocity increases proportionally with substrate concentration. As substrate concentration increases, more enzyme active sites are filled. Reaction rate continues to rise until all active sites are occupied. Stage is eventually reached where all enzyme molecules become saturated with substrate. No further increase in reaction velocity occurs despite increasing substrate concentration. This maximum possible rate is called Vmax, maximum velocity. The curve obtained when reaction rate is plotted against substrate concentration is a rectangular hyperbola. In Michaelis-Menten equation, Leonard Michaelis and Maud Menten proposed a model to explain enzyme kinetics based on the formation of an enzyme-substrate complex. So Km is defined as the substrate concentration at which reaction velocity is half of Vmax, an indicator of enzyme affinity for its substrate. In that, we can also see the Lineweaver-Berg plot. Since Vmax is difficult to measure experimentally, Lineweaver and Berg developed a plot using the inverse form of the Michaelis-Menten equation. Produces a straight line which enables easier calculation of Km and Vmax. So what are all the factors affecting enzyme action? So the rate of enzyme catalyzed reaction is influenced by several factors, including temperature, pH, enzyme concentration, and substrate concentration. These factors determine how efficiently an enzyme converts substrate into product. We can see that in detail in the further slides. And now we can look into the enzyme inhibition. Enzyme inhibition refers to the decrease or loss of enzyme activity due to the action of specific chemical substance known as inhibitors. These substances interfere with normal enzymatic function and can provide valuable information about enzyme specificity, active site structure, and catalytic mechanism. Some medicinal drugs act as the enzyme inhibitors, while others are toxic and can permanently damage enzymes by interfering with metabolic reactions. So, what are all the types of enzyme inhibitors? There are two types, irreversible inhibitors and reversible inhibitors. In irreversible inhibitors, these inhibitors bind permanently to the enzyme. They destroy or modify essential functional groups in the enzyme molecule. This causes permanent loss of catalytic activity. Example, toxic heavy metals and chemicals, arsenic, lead, mercury, and insecticide. Even at low concentration, such substance can precipitate or permanently deactivate enzymes. Reversible inhibitors. These inhibitors do not perform permanent bonds and their effect can be reversed. They are useful for studying enzyme structure and reaction mechanisms. Reversible inhibitors are of two main types, competitive and non-competitive inhibitors. These inhibitors closely resemble a structure in substrate. In competitive inhibition, the inhibitors compete with the substrate for binding to the active site of the enzyme. When bound, they prevent the substrate from binding but do not undergo reaction or form product. Increasing substrate concentration can reverse the inhibition. Example, malonate, which structurally resembles succinate. It inhibits succinate dehydrogenase by occupying the active site. Increasing succinate concentration overcomes inhibition. Non-competitive inhibition. These do not compete with the substrate for the active site. They bind at a different site on the enzyme, which is known as allosteric site. Binding changes the conformation of the enzyme, thereby distorting the active site, reducing catalytic activity. They may bind to either free enzyme substrate complex. Increasing substrate concentration does not restore activity. Example, L-isoleucine inhibits L-threonine dehydratase through non-competitive inhibition. Non-competitive inhibitors are also naturally used by cells for regulating metabolic pathways. In uncompetitive inhibition, we can see that inhibitor forms a permanent covalent bond with the enzyme. Thank you everyone for your active participation and attention throughout the session. I hope this introduction to biochemistry and enzymes has helped you understand how these fascinating molecules play an essential role in the chemical reactions taking place within our body. I look forward to exploring the concepts of enzymes, their mechanism of action, and their importance in nutrition and health with you in the upcoming sessions. Thank you and have a wonderful day.